Hi everyone, welcome back to the channel. Today we'll be discussing about mRNA vaccines. What are mRNA vaccines? An mRNA vaccine is a new type of vaccine that uses messenger RNA or mRNA to teach our cells how to make a harmless protein from a pathogen. This protein then triggers an immune response. Unlike the traditional vaccines, mRNA vaccines they do not contain weakened or inactivated viruses. Instead, they provide genetic instructions that allow our own cells to produce the target protein and train the immune system safely. How are mRNA vaccines made in the laboratory? mRNA vaccines are made and produced using a fast cell-free process called in vitro transcription or IVT. First, the scientists design a DNA template containing the genetic code for the target protein. This DNA is then amplified and purified. Next, the DNA is linearized and used as a template to synthesize mRNA using RNA polymerase and nucleotide building blocks. The newly produced mRNA is then modified with a 5' cap and a poly-A tail, which will improve its stability and allow efficient protein production. After the synthesis, the mRNA is purified to remove unwanted DNA, enzymes, and reaction components. Finally, the purified mRNA is packaged into lipid nanoparticles, which are tiny fat-based carriers which protect the mRNA and help it deliver into human cells. How do mRNA vaccines work? After vaccination, the lipid nanoparticles which carry the mRNA into the cells, inside the cytoplasm, the ribosomes will read the mRNA instructions and produce the target protein, such as the viral spike protein. The immune system recognizes this protein as foreign and activates both antibody producing B cells and protective T cells. This creates immune memory, allowing the body to respond rapidly if it encounters the real pathogen in the future. The mRNA itself is temporary and it is naturally broken down by the cell within a few days. What are the key characteristics of mRNA vaccines making it so special and successful? First is its extreme speed and scalability. mRNA vaccines can be designed and manufactured much more faster than traditional vaccines. If a pathogen changes, the scientists only need to update its genetic sequences. Its high safety and precision. mRNA never enters the nucleus, which is the area where the DNA is, and it cannot enter or alter the DNA. So it will only provide instructions for making a harmless target protein and is then quickly degraded after use. Third is the strong immune response. mRNA vaccines, they stimulate both antibody production and T cell responses, providing broad and effective protection against disease. Why is mRNA specifically used? mRNA offers several advantages that make it ideal for vaccine development. First, it works directly in the cytoplasm, which is the area in the cell where protein synthesis occurs. So it does not enter the nucleus. Second, it's naturally temporary. After the protein is produced, the mRNA is rapidly degraded by normal cellular process. Third, mRNA is more efficient than DNA-based approaches because it skips the additional transcription steps which are required inside the nucleus. Finally, because our own cells produce the protein, it is folded and it is processed naturally, so it results in a more authentic immune response. Now let's discuss what are the four main biomedical applications of mRNA technology. First is infectious disease vaccines. This is the most successful application of mRNA technology. COVID-19 vaccines, like those developed by Moderna and Pfizer, have demonstrated how quickly effective vaccines can be produced during a global pandemic. Second is cancer immunotherapies or oncology vaccines. So, personalized mRNA vaccines are being developed to help the immune system recognize and attack cancer cells by targeting specific tumor-specific antigens. Third is protein replacement therapies. mRNA can provide instructions for producing missing or defective proteins, offering potential treatments for several rare genetic disorders. And finally, gene editing and cell therapies. Researchers are using mRNA to deliver gene editing tools like CRISPR and to engineer immune cells for advanced therapies, which include the next generation cancer treatments. Finally, concluding, mRNA technology has transformed the modern medicine by giving a fast, flexible, and highly precise platform for treating disease. From vaccines and cancer therapy to protein replacement and gene editing, mRNA is expanding far beyond infectious disease and it is shaping the future of personalized medicine. I hope you guys like this video. Please like, share, and subscribe for more such videos. Thank you, everyone.